Masikini moyo wangu umefanya nyama kwa bucha unateketea.
Welcome back to my channel. Now continue from my video about genetic engineering part 1. Okay, in part 2 we want to discuss more detail about the vectors, the host cell, and example of genetic engineering, which is to produce insulin by genetic cloning. Insulin. This insulin will inject by this fan, okay, and it will inject in our body, and the insulin will enter our bloodstream. Okay, so how this insulin is produced? So in the next step of this video, we will see the steps of gene cloning, how we produce insulin from gene cloning, and we use cDNA as donor DNA. Okay, let's see the steps. Okay, bagaimana insulin ini dapati daripada proses cloning? Okay, kita lihat, kita dah tahu, sudah mengetahui ciri-ciri vektor yang digunakan dan juga gene DNA yang digunakan. Okay, untuk insulin yang ini kita boleh gunakan sama ada dua cara, menggunakan genomic DNA atau gene DNA, atau menggunakan complementary DNA. Okay, so this one menceritakan tentang insulin dulu dapat straight daripada genomic DNA animal. Okay. Okay, now onwards, untuk menghasilkan insulin kita menggunakan genetic engineering, okay, dan kita menggunakan cDNA, that means complementary DNA that copy DNA gene from DNA donor. Okay, so the bacteria mostly used is E. coli bacteria. Okay, so the first step is isolation the donor and cloning vector. Okay, so the donor, okay, either synthetic gene or is it a generic DNA or cDNA, which is a copy of coding sequence of the human gene. Okay, kita ulang kaji semula. Okay, this synthetic gene, that means using nucleotides and joining them together in right order. Okay, for cDNA, composed of coding and non-coding, there is a copy of the coding sequence is called cDNA. So the process we have known already before this, that produced from the reverse transcription of mRNA. So they will produce the cDNA. Okay, let's see the step. Okay, the first of all, in step of isolation, we after the isolation we must cut the DNA. Okay, so in this process we use cDNA. So we cut the cDNA with restriction enzyme. Okay, and see that they produce the sticky end. Okay, dia menghasilkan hujung yang sticky. Cloning vector yang digunakan adalah plasmid. Okay, plasmid yang dapati daripada bacteria E. coli. Okay, this plasmid contain the marker, marker that we say amphiphylline. Okay, where the bacteria that have this plasmid, okay, will dia akan dibiarkan dalam medium ada ampicillin. Okay, ampicillin adalah sejenis antibiotik. Okay, dan untuk plasmid ini juga dia ada satu restriction site. Okay, kalau ikutnya plasmid boleh ada lebih dari satu restriction site. Okay, so the restriction site is at the LATZ gene. Okay, which LATZ gene is used to produce a beta-galactosidase. And if it take the fragment, the DNA, the LATZ will not function. Okay, remember the before step in part 1 where we already know the basic step of cloning, where we already know about the function of LATZ. Okay, so the first one, okay, isolation. Okay, so we use the cDNA. Okay, and then after isolation is the restriction. Okay, and then after restriction is the ligation process. Okay, where the DNA ligase enzyme is used. Okay, and then we Okay, this one is a plasmid that contain the recombinant DNA after process ligation. Okay, in transformation, okay, there are the plasmid will transform into the host cell. Okay, when we see the host cell, bacteria, some take the plasmid, then some does not take the plasmid. Okay, so yeah, which the bacteria that does not take plasmid will die. Okay, yang tak ambil plasmid die. Yang plasmid ni pula kita perlu melalui proses screening. Okay. It's the bacteria will grow in the medium that contain X gel and ampicillin. And remember, the blue colony, which does not contain the plasmid because the lead Z is function. So the lead Z function, it will change the X gel color to blue. Okay, so the color blue is not taken the plasmid, and the white colony is taken the plasmid. So the white colony will go into the fermentation to produce a large number of insulin. Okay,